back, ladies and gentlemen. We're going to look at some DNA mutations. So we've talked a lot about how DNA codes for proteins, and it's a very complex process. Wouldn't you agree? Of course you would agree. It's a complex process. And one thing we need to understand is what happens when things aren't coded properly or something in the DNA changes. How does that impact the proteins that are being produced? And that's what we're going to look at today, are some DNA mutations. So hopefully by the end of this lecture, you'll be able to understand and explain the differences between different types of mutations in DNA. Some mutations are more harmful than others, and we need to understand those differences and to be able to predict the effects of a mutation on the synthesis of a protein. So if we do have a mutation, what causes it, and what effects are that going to have on the protein being made? If it's a life-essential protein, odds are the organism cannot survive, or the cell cannot survive. If it's not essential, there may not be very many harms associated with it. So let's take a look at some of these DNA mutations that we'll be looking at today. First off, the term mutation just simply refers to a change in DNA. So anytime you have some form of mutation, it's just an A, T, C, or G has been changed within the molecule of DNA inside of the cell. And this can result from many different factors. One of them is replication mistakes. The process of replication or protein synthesis isn't perfect every time. So there are some mistakes that may be made, and those mistakes can be very detrimental or damaging to the cell if it's in an important part of the DNA. Additionally, mutagens, or factors that can cause mutations, have an impact on them as well. And these are things like UV rays. You've probably heard about tanning and being out in the sun giving you skin cancer, and that's true because the UV rays can actually penetrate into the cells in your skin and alter the DNA. Pollution can cause problems as well. Things like radiation can be problematic as well. So lots of different environmental factors can have an impact on mutations that occur inside of your DNA. Now, a lot of cells have mechanisms to prevent mistakes and even repair them. If a cell has a mistake, a lot of times the cell will simply self-destruct in a process called apoptosis. But this doesn't always happen, and not all of them are repaired. And this can cause some serious problems with the cell, including cancer. A change in DNA can result in a change in the protein. So for example, if we have a codon that is changed from UAG to AAG, that U ends up getting genetically altered to an A, the amino acid then changes from a stop codon to lysine. And this can significantly alter a protein because that amino acid sequence is very specific to that protein. And if that amino acid sequence changes, well, then the protein is going to change. And if it's a life-essential protein, that cell cannot survive. Additionally, a change in DNA may not necessarily change a protein at all. For example, the codon going from UAU to UAC, well, if we look at the coding chart, that is still lysine for both. So this would not result in a change in the protein at all. So some mutations can be damaging to the cell and others may not change anything at all. It just depends on where the mutation takes place inside of the DNA. All right? So some may cause changes in proteins and some may not. Some may be deadly to the cell and some may not as well. Just keep that in mind as we go through this. Several different types of mutations. One is known as a substitution, and that could simply just change one amino acid. So we look at the mRNA sequences below. We have UAUC. That can be genetically changed for one reason or another to UAUG. Now this will just affect one amino acid. Every amino acid afterwards may not necessarily be affected by it because you see the rest of the sequence stays the same. A frameshift mutation alters every amino acid after the change. So you can imagine that this is much, much more damaging to the cell than that of a substitution mutation. Because you remember that amino acids are made from codons, 3 nucleotide sequences. So if we look, UAU is the same for all 3 of them down there, the original and then the 2 below that have a mutation. But if we insert a C, all of the codons, the 3 nucleotide sequences after the insertion or deletion are altered. So instead of UAU, CAC, GAG, CCG, it's UAU and now it's CCA, CGA, GCC with an extra G at the end. So that impacts every single codon after the insertion. Same thing with the deletion. It has an impact on every single codon after that deletion. So frameshift mutations are much more dangerous because it alters every codon after that particular mutation, which could alter every amino acid after the mutation. Now, some mutations can be inherited and some cannot. So it's important to understand the difference between these two. Mutations may not even necessarily cause a disease or disorder if it's in a non-coding region of the DNA, such as an intron. Remember, those are spliced out, so those aren't actually read by the ribosome when protein synthesis takes place. Some mutations don't even cause a change in amino acid, if we remember the lysine example from before. Mutations in body cells cannot be passed to offspring, something really important to understand. So if you get skin cancer from ultraviolet rays, you aren't going to pass that skin cancer onto your offspring because that's in a body cell. The only cells that are passed on to the offspring, the only DNA, is that in which that are found in sex cells, such as sperm or egg. So if there are genetic mutations in your sperm or egg, those can be passed on to your offspring. Body cells and mutations there cannot, but sex cells can. And that's something that we'll talk a little bit about when we get more into the genetics part of DNA. So we look at here at just some of the mutations, and I just want you to understand how simple mutations can have an impact. We look at the point mutations below, and we look at the mutations that can be caused there. So you see from the original sequence, we changed that codon in the middle there from CAA to CAG, and that really doesn't have an impact because CAA and CAG code for the same amino acid. But if we look at the one underneath it, UCU is now CCU. That changes the amino acid, which would then change the protein. Additionally, we could even change something more dramatic. For example, that second code, UAU, could be changed to UAA. So instead of tyrosine, it codes for a stop codon. So it would stop reading the sequence, and all the amino acids afterwards wouldn't get read. So you can see that some mutations can be and are more harmful than others. Hopefully you understand a little bit about the difference between the different types of mutations in DNA, and you're able to predict the effects of a mutation, whether frameshift or point mutation, on the synthesis of a protein. Again, guys, remember to take your video quiz, and we'll talk to you later. Have a good day.